First, we will briefly discuss the role and composition of the immune system. Our body is constantly exposed to various pathogens and other harmful materials. In some cases, people develop tumors. It is the primary role of the immune system to counter these pathogens or cancers and keep our body healthy. The immune system comprises different immune cells. These include neutrophils, macrophages, and dendritic cells, which are phagocytic. They engulf and digest bacteria invading the host cells. In addition, T or NK cells kill virus-infected and cancer cells. Antibodies secreted from B cells bind to pathogens and tumor cells to remove or detoxify them. Among these immune cells, T cells attack cancer or infected cells directly. As shown in this slide, T cells recognize these infected cells as dangerous, and this recognition activates them to attack and kill their targets immediately by secreting cytotoxic factors. This slide shows how T cells recognize target cells. Each T cell has a receptor on its cell surface called the T cell receptor, TCR. Every cell, including cancer cells, expresses an antigen and MHC molecule complex on its surface. Each peptide MHC class I complex is composed of an alpha chain, non-MHC beta 2M, and antigen. In the case of cancer cells, the antigen peptide loaded on the MHC is a fragment of multiple proteins expressed inside the cell. TCR recognizes each individual complex and assigns cellular identity. Monitoring antigen-specific T cells is very important and helps clinicians to evaluate the status of various diseases or patient response to treatment. The question that next arises is the following: how can we detect antigen-specific T cells? To answer this question, we will now discuss MHC Tetramer technology in more detail. Since MBL has more than 40 years of experience in developing antibodies, we decided to use this expertise to develop novel tools to evaluate TCRs. Initially, we focused on developing specific antibodies to target each TCR. However, the nature of TCRs made constructing a specific antibody difficult, and we encountered challenges, such as the preparation of proper immunogens and a high degree of similarity among different TCRs. Consequently, antibodies against individual TCRs had very high cross-reactivity with other TCRs. Thus, this approach was too complicated and time-consuming.